I'm Harry. I worked in Peter Dearden's lab, and I was studying the use of the big-headed ant, Pheidole megacephala, as a genetic model for other hymenopterans. Invasive hymenopterans in New Zealand pose a critical threat to our biodiversity and agriculture. There are management strategies in place for wasps like the yellow jacket or vespula, where they're poisoned quite big numbers, but these management strategies haven't seemed to work very well for the European paper wasp, Polistes dominula, which has become widely established throughout the South Islands. And the fact that these management strategies don't work makes this wasp a primary target for novel genetic biocontrol. However, the husbandry of these wasps is very difficult due to their complex life cycles and specialized diets. So my goal was to evaluate the use of the big-headed ant, Pheidole megacephala, as a manageable genetic model for these hymenopterans, as ants are much easier to scale and they also share highly conserved evolutionary history and genomes with eusocial wasps. Oh. So to test the theory that these wasps were hard to grow in the lab and care for, we collected many different colonies from the wild in Nelson and Wanaka, and we housed them in different cage types to test for different variables. Alongside these wasps, we maintained the P. megacephala colonies in test tube setups and formicaria with controlled humidity. A big part of being a model organism is the ability to uptake different genetic elements. And so to test for this in the ant, we fed them a plasmid with the green fluorescent protein that was encased in nanoparticles, and we put this in their feed. To test for the uptake, we used DNA extraction and PCR. And to test for the expression of the plasmid, we used fluorescence microscopy. What we found was that, in fact, the wasp colonies did show stagnant or declining growth that plateaued at less than 40 days, as this is the life cycle from egg to adult of these wasps, which means that any growth we did see was due to the presence of eggs that were already in the nest before we collected them. And it's unlikely that they laid eggs, and if they did, these eggs did not reach adulthood. Conversely, the ant colonies we had successfully produced elates, which are individuals capable of mating, and these did mate in the lab, so we got more colonies from them. What we found is that there was natural autofluorescence in the larvae, which masked the detection of the GFP, as you can see in that top image, both larvae expressing a green color, both the control and the experimental. But if you look at the social stomachs, the next one down, you can see that the experimental does have the blue dye, which means they are ingesting the plasmid. So next time we would need to use a fluorescent protein that fluoresces in a different wavelength than these larvae do. To test for the uptake properly, we used the PCR. And if you look at the PCR, you can see a second experimental worker, and both experimental larvae showed a band at around 350 base pairs, which is what we would expect from presence of the plasmid. But we can also see that there is another band in the negative control, which means that there was likely some contamination during our experiment. So another experiment would have to be run from the start to get a more concrete evidence. We also attempted to get a sequence of the ants, and we achieved high quality with 11.65 gigabases of data and a high N50, which means that we have high quality DNA that is really good groundwork for finding out how much conservation there is between the ant and our target wasp in their genomes. All of this together is a great base groundwork for the development of these ants as a genetic model. Thank you very much. I want to also say thank you to my supervisors, Hamish and Kim and Peter and everyone in the Dearden lab. It was great fun. Had a blast.
Thank you. Does anyone have a question for him?
Well, thanks, Harry. I like the idea of using ants as models, as systems for Hymenoptera. I guess one issue with the big-headed ant is that it has quite a clear caste system with kind of soldiers and workers and quite a bit of size variation amongst individuals. Is that going to cause complications when trying to compare it to wasps, or is that an advantage?
It's not necessarily an advantage, but the caste differentiation in these ants is not actually determined by their genome, so that all of the ants will show the same genes, pretty much the same genes. So
Okay.
it shouldn't have effect.
Thanks.